Around 7 million years ago, a slow and silent shift began, one that would eventually set our ancestors on a different path from chimpanzees and bonobos. There was no single moment, no clear line, but over countless generations, the first early hominins emerged. But who were the first humans to walk that path? What did they look like, and how did they survive? To find out, we must travel back millions of years to the story of our earliest ancestors, the forgotten pioneers of humanity. We must begin at the very roots of our evolutionary journey, deep in prehistoric Africa, where the earliest members of our lineage first emerged. The oldest known species in the human family tree is considered to be a hominin named Sahelanthropus Tchadensis, a species that lived sometime between 7 and 6 million years ago in West Central Africa. The fossils, all found at the site of Todos Manala in the Jurab Desert in Chad, include several jaw pieces, teeth, and a small but relatively complete cranium nicknamed Tumai, which means hope of life in the local language. Although we have only cranial material from Sahelanthropus, studies so far show this species had a combination of ape-like and human-like features, albeit more ape-like. Ape-like features included a small brain, estimated to be between 320 and 380 cubic centimeters, which is even slightly smaller than a chimpanzee's, a sloping face, very prominent brow ridges, and an elongated skull. Human-like features included small canine teeth and a short middle part of the face. The enamel thickness of the teeth was intermediate between that of living apes and Australopithecines, a group of later hominins who lived from 4.4 to 1.9 million years ago, suggesting a transitional dental adaptation. Importantly, there was no lower jaw diastema, a gap space between teeth in the lower jaw, usually between the canine and the adjacent incisor or premolar, typically found in other primates, indicating a closer dental structure to later hominins. The cheek teeth were also comparable in size to those of the Australopithecines than to the great apes, providing further clues to its dietary habits and evolutionary relationships. The conclusion made from this is that their diets were primarily plant-based. It is predicted that Tumai mostly consumed food such as leaves, fruit, seeds, roots, nuts, and insects. We are fairly confident that Sahelanthropus could walk upright, based on the position of the foramen magnum, the large opening where the spinal cord exits the skull. In Sahelanthropus, it's located further forward on the underside of the cranium than in apes or any other primate except humans. This feature indicates that the head of Sahelanthropus was held on an upright body, probably associated with walking on two legs. However, as no leg or foot bones have been found, we can't confirm how well it walked on two legs. Most scientists think Sahelanthropus was a tree climber that also walked upright sometimes, likely walking bipedally between trees on the ground, but still spending most of its time on all fours in the trees, much like chimpanzees today, but likely with an upright posture better suited for walking. No evidence of cultural attributes are linked to Sahelanthropus, but this species may have used simple tools similar to those used by modern chimpanzees, including unmodified stones or sticks and other plant materials that were easily shaped. The ancient environment where the fossils were found consisted of lake, forest, river, and wooded savanna. It shared this environment with an array of majestic animals. Thousands of vertebrate fossils have been found at the site, including elephants, giraffes, antelopes, hippopotamus, crocodiles, lizards, monkeys, fish, and wild boar. Alternative interpretations of Sahelanthropus Tchadensis suggest that this ancient hominin may hold various positions in the evolutionary tree. One interpretation posits that Sahelanthropus could be a common ancestor of both humans and chimpanzees, providing a crucial link in our understanding of hominin evolution. Another perspective suggests that while Sahelanthropus Tchadensis is related to both humans and chimpanzees, it is not a direct ancestor of either lineage, indicating a more complex branching of the evolutionary tree than previously thought. Additionally, some interpretations propose that Sahelanthropus Tchadensis may actually be an ancestor of gorillas, challenging the conventional placement of this species within the human evolutionary lineage. Overall, Sahelanthropus Tchadensis, or Tumai, marks a profound chapter in the story of human evolution. Positioned near the root of our family tree, Tumai serves as a crucial link between apes and humans, offering rare insight into the earliest steps on the path that would eventually lead to us. But before we go ahead and talk about further early hominin species, one fundamental question remains. What drove these early hominins to leave the safety of the trees and start spending more and more time on the ground? This major shift is thought to be the result of both environmental changes and genetic adaptations. These changes led to different evolutionary paths, with one lineage adapting to life on the ground and eventually developing bipedalism, while the other remained primarily tree-dwelling. While traditional theories often place the origins of human bipedalism on the savanna, recent research suggests that it may have first evolved in the trees. Studies on chimpanzees show they frequently use bipedal locomotion in trees, particularly when reaching for food. This challenges the idea that bipedalism was solely an adaptation to open grasslands and suggests that tree dwelling may have played a crucial role in its development. Bipedalism in trees may have initially evolved as a feeding posture, allowing early humans to reach food on branches more effectively. While the shift to more open habitats definitely influenced the evolution of bipedalism, the research suggests that arboreal adaptations were also crucial in shaping this trait, giving it a head start when the environment did become more open. Around the time of the split, around 6 to 8 million years ago, Africa experienced significant environmental changes, including a shift from dense forests to more open woodlands and savannas. This was because Earth's overall climate began to cool and dry during this period, which is linked to changes in ocean circulation and atmospheric CO2 levels. This was part of a long-term trend that began in the Miocene epoch. As temperatures dropped, rainfall decreased in many regions, including Central and Eastern Africa. Tropical rainforests, which need high moisture, began to shrink and were replaced by more open habitats like woodlands, bushlands, and savannas. Walking on two legs offered advantages in these new environments, such as better visibility over tall grasses, increased efficiency in traveling long distances, improved thermoregulation, increased foraging efficiency, and the ability to carry tools and infants. Over millions of years, these changes in habitat and locomotion led to further genetic and physical divergence between the human and chimpanzee lineages. The split was not a clean break, but rather a gradual process with some interbreeding possible between the two lineages during the early stages of divergence. The second oldest human ancestor after Sahelanthropus Tchadensis is considered to be Orrorin Tugenensis, the only species classified in the genus Orrorin. The age of the specimen has been estimated to 6 to 5.8 million years. Presently, 20 fossils have been found at 4 sites in the Tugen Hills of Kenya. These remains include parts of the lower jaw in 2 pieces, the front part of the chin area, several individual teeth, 3 pieces of thigh bones, part of an upper arm bone, a finger bone, and the tip of a thumb bone. Orrorin was still probably quite chimp-looking, but natural selection had already begun shaping a body that showed early steps toward the modern human form. Its thigh bone shows signs of bipedalism. The femur head was rounded, the neck was long, and a key muscle attachment, the lesser trochanter, pointed inward, all features linked to walking upright. However, other bones suggest it still also climbed trees. One finger bone was shaped like a human's and has been linked to tool use, though in this case it may have been more about strong gripping for climbing. The brain size was probably similar in size to a modern chimpanzee, but lack of cranial material makes this difficult to determine. Orrorin had relatively small teeth for its body size. Its canines were still ape-like, but smaller, similar to those in Miocene apes and female chimpanzees, but the back teeth were small, like those of modern humans. Orrorin lived in dry evergreen forests, further showing that bipedalism may have evolved in wooded environments, not just grasslands and savannas. From Orrorin's low, rounded molars and small canine teeth, paleoanthropologists can infer that this species ate mainly a plant-based diet. This probably included leaves, fruit, seeds, roots, nuts, and insects. Like Sahelanthropus Tchadensis, Orrorin is at the base of the human family tree, but has more ape-like features than human-like ones. Some experts think this species may have even lived before the ape and human line split, so could be an ancestor of both lines, or that it was a basal member of the hominin clade. Another hominin species that lived in and around the time of the split between humans and chimps is the genus Ardipithecus, which two species are described in the literature. Ardipithecus Kadabba, dated to approximately 5.6 million years ago, and Ardipithecus Ramidus, dated to about 4.4 million years ago. Ardipithecus Kadabba is comprised of 11 specimens from 5 localities in eastern Africa in the Middle Awash region of Ethiopia, discovered between 1997 and 2000. They represent at least 5 individuals and include teeth, jaws, hand, toe, arm, and collarbones. From these fossils, we know Ardipithecus Kadabba was similar in body and brain size to a modern chimpanzee, but was bipedal. It had canines that resembled those in later hominins, but the front teeth still stuck out like an ape's. One bone from the large toe has a broad, robust appearance, suggesting its use in bipedal push-off. Instead of eating mostly fruit and soft leaves like chimpanzees and Orrorin and Sahelanthropus, there is evidence that Ardipithecus Kadabba ate a variety of fibrous foods. Scientists theorize this because the back teeth of Ardipithecus Kadabba are larger than a chimpanzee's, but its front teeth are narrower. This evidence suggests this species did most of its chewing in the back of its mouth. This type of chewing would focus on hard-to-eat foods like fibrous nuts. Fossil evidence from the site indicates the area was a mosaic of woodland and grasslands with lakes, swamps, and springs. The discovery that this species lived in a forest environment as well as open grasslands further complicates the theory about what kind of environment fostered the evolution of bipedalism. Did bipedalism evolve to take advantage of new open grassland environments, as was once believed, or did it first evolve in the trees? Or could it have been shaped by both environments? Ardipithecus Kadabba is suggested to have directly given rise to Ardipithecus Ramidus, a species that lived about 4.4 million years ago. It is known from the partial skeleton nicknamed Ardi. This species gives us an incredible glimpse into early human evolution. Ardi's bones, including parts of the skull, teeth, arms, pelvis, legs, and feet, reveals once again a species with a mix of traits seen in both apes and later humans. The main traits that link Ardipithecus Ramidus to later hominins include small, blunt canine teeth which had little difference in size between males and females. This suggests a change in behavior, possibly less aggression. The skull was small-brained like a chimpanzee's, but lacked the strong jaws seen in later species adapted to heavy chewing. It also didn't have the large front teeth used for eating lots of fruit, as seen in chimps. A forward-positioned opening at the base of the skull suggests upright posture. The pelvis also showed signs of bipedalism, especially in the upper part, allowing for upright walking. However, the lower pelvis and limbs still showed strong adaptations for climbing, suggesting Ardi still spent much of her time in the trees. It had elevated shoulder joints for reaching up for branches, an adaptation suited for climbing, and suspensory behavior unlike modern human shoulder joints, which are positioned differently to support long-distance walking, the throwing of objects and weapons, and tool use on the ground. Ardi also had long arms and long curved fingers, mobile wrists, and a poorly differentiated thumb, meaning that they had poor opposability. Orangutans are the only great ape with a similar thumb morphology and hence have poor manual dexterity. The feet had a divergent big toe, meaning it stuck out to the side, similar to chimpanzees, and was adapted for grasping branches, indicating an arboreal lifestyle. However, it also possessed a unique small bone within a tendon that likely provided more strength and stability for upright walking, and the hands lacked features for knuckle walking, suggesting a transitional form between tree-dwelling and ground-dwelling locomotion. This unique combination shows that Ardi could move in the trees but also walk on 2 legs, though not like modern humans. Associated animal and plant fossils indicate this species lived in a relatively moist and heavily forested woodland. Fossil species include plants such as fig, palm and hackberry, and animals like columbine and baboon-like monkeys, kudu, peafowl, bats, shrews, rodents, doves, eagles, owls, and parrots. They were probably more omnivorous than chimps, based on the size, shape, and enamel of the teeth, and fed both in trees and on the ground. Their diet may have included nuts, fruit, tubers, insects, small vertebrates, and eggs. The evidence is inconclusive, but studies suggest the front teeth were regularly used for clamping and pulling, possibly reflecting a diet that also included a large amount of leaves. Carbon isotope studies of teeth show they ate woodland rather than grassland plants, suggesting that Ramidus lived and foraged primarily in woodland environments, not open grasslands. The discovery of Ardi was very important within the scientific community at the time, as prior to the find, most people reasonably, but incorrectly, assumed that the last common ancestor of chimpanzees and humans was very much like the modern chimpanzee, a frugivorous knuckle-walking primate largely restricted to an African tropical forest habitat. The Ardipithecus fossils have no specifically chimpanzee-like features, and this indicates that the earliest hominids never passed through a chimpanzee-like stage. This finding surprised many scientists and much of the public, who had long envisioned that the ancestors of humans were early chimpanzees. Following Ardipithecus, the next major group of hominins in the fossil record is the Australopithecus species. Australopithecus included several species, such as Australopithecus Afarensis, made famous by the Lucy fossil, as well as at least 8 more. They are known from a series of fossils found at numerous sites in eastern, north-central, and southern Africa. The various species of Australopithecus lived 4.4 million to 1.9 million years ago. The Australopithecines possessed a more gracile and less robust body than their predecessors, and they are the earliest hominin ancestors that show definite habitual evidence of bipedalism. While the Australopithecines were definitely bipedal, many scientists believe that tree climbing was one behavior that was probably retained by all early hominins, including the Australopithecines, until the appearance of the first Homo species about 2.5 million years ago. A set of footprints found in Laetoli, Tanzania, shows where early hominins once walked along the shore of an ancient lake around 3.6 million years ago. These footprints were preserved in volcanic ash and most likely belonged to Australopithecus afarensis. The hominin prints were produced by 3 individuals, one walking in the footprints of the other, making the original tracks difficult to discover. The footprints demonstrate that the hominins walked upright habitually, as there are no knuckle impressions. From these footprints, we can tell, for the first time in hominin evolution, the feet had developed a modern human-like arch, rather than the flatter feet of apes and earlier hominins, and the big toe aligned with the other toes. But the feet also retained some flexibility in the joints, and they still possessed curved toe bones, suggesting continued climbing ability. Evidence suggests these hominins seem to have moved in a leisurely stroll and may not have yet been capable of running at a faster pace. Australopithecus Anamensis, the oldest Australopithecine, existed about 4 million years ago and has a mixture of primitive features in the skull and advanced features in the body. The upper end of the shin bone shows an expanded area of bone and a human-like orientation of the ankle joint, indicative of regular bipedal walking. Long forearms and features of the wrist bones suggest these individuals probably climbed trees as well. The elbow joint was more human-like in being relatively flexible, rather than the more rigid, locking elbows that quadrupedal apes have to support their bodies as they move about. The cranium combines some more ancestral features, such as a protruding face and a long and narrow brain case, with some more derived features, such as forwardly projecting cheekbones. Australopithecus Anamensis individuals had thickly built, long, narrow jaws, with their side rows of teeth arranged in parallel lines. Their strong jaws, combined with heavily enameled teeth, suggest Anamensis individuals may at times have eaten hard, abrasive foods, but they likely were plant eaters in general, relying on both fruits and tough foods such as nuts. There was an enormous difference between the body sizes of males and females, what is known as sexual dimorphism. This difference between the sexes in body size was similar in degree to that shown by modern gorillas and orangutans. The strong physical differences between males and females, along with certain features in the teeth, suggest that this species likely had a social structure involving multiple mating partners. There are 2 main possibilities. 1. A polygynous system where one male mates with several females. Like in gorilla groups, in this setup, both males and females usually leave the group they were born into. Or second, a polygynandrous system, where multiple males and females live and mate within the same group. This is seen in chimpanzees and bonobos. In their case, males stay in the group they were born in, while females leave when they become adults. If we use chimps and bonobos as a model for early human ancestors, the second option, a multi-male, multi-female system, is more likely. Plant and animal fossils and the analysis of ancient sediments and rocks provide clues about this species' environment. They lived near an ancient inland lake that existed in the basin where Lake Turkana is now found. The region was subject to volcanic activity, and various volcanic eruptions produced layers of volcanic ash that covered the ground. The fossilized remains of this species were found trapped between different layers of ash, and this has enabled this species' fossils to be reliably dated to between 3.8 and 4.2 million years in age. These early ancestors lived in forests and woodlands that grew near the lake. The Australopithecus species who came after Anamensis was Australopithecus Afarensis, Lucy's species, which lived between 3.9 and 3 million years ago, primarily inhabiting modern-day Ethiopia, Kenya, and Tanzania. The Lucy fossil is one of the most remarkable discoveries in paleoanthropology. It is a relatively complete skeleton, about 40% intact, that has helped scientists to draw a clearer picture of what these early human ancestors looked like. Many cranial features were quite ape-like, including a low, sloping forehead, a projecting face, and prominent brow ridges above the eyes. Their brain was still small, averaging approximately 430 cubic centimeters and comprised about 1.3% of their body weight. But reorganization of the brain may have begun with some enlargement to parts of the cerebral cortex, which plays a role in thinking, memory, decision-making, sensation, and voluntary movement. A small hyoid bone which helps anchor the tongue and voice box found in a juvenile specimen suggests Afarensis had a chimp-like voice box, confirming they would not have been able to speak. The semicircular ear canal was similar in shape to African apes, suggesting this species was not as fast or agile on two legs as modern humans. Their jaws and teeth were intermediate between those of humans and apes. The jaws were relatively long and narrow. In the lower jaw, the teeth were arranged in rows that were slightly wider apart at the back than at the front. In the upper jaw, the placement of the last molar results in tooth rows that curve in at the back. Sexual dimorphism still existed within the species. Females grew to only a little over 1 meter in height, and males were much larger at about 1.5 meters in height. The pelvis and leg bones far more closely resemble those of modern man and leave no doubt that they were bipedal, although adapted to walking rather than running. Limbs displayed human-like features that also indicate an ability to walk on two legs. This includes their thigh bones that slanted in toward the knee, arched feet, and wide heels, and their big toes aligned with the other toes and were not opposable. Ape-like features that suggest an ability to climb trees include powerful arms with long forearms and shoulder blade sockets that face upwards like an ape's rather than to the side like a human's, but it did share other similarities with human shoulder blades. The finger and toe bones are curved and proportionally longer than in humans. Most scientists consider this evidence that Afarensis was still partially adapted to climbing in trees. Others consider it evolutionary baggage. However, the big toe appears to lack the grasping ability common to apes, and the hands are similar to humans in most other details, such as relatively short fingers and a more refined thumb, which would have allowed for precise manipulation, though not yet to the same degree as modern humans. Microscopic analysis of their tooth enamel shows that they mostly ate fruits and leaves rather than nuts and other hard plant material. Their cone-shaped ribcage indicates they had large bellies adapted to a relatively low-quality and high-bulk diet. The position of the sagittal crest toward the back of the skull indicates that the front teeth processed most of the food. This species occupied a wide range of environments. Some populations lived in dense forests, near lakes and rivers, while others inhabited open savannas or sparsely wooded areas. This ecological flexibility is significant, as Australopithecus afarensis appears to be the first hominin in the fossil record to have adapted to life in the open savanna. Living in such varied habitats would have exposed them to a broader range of resources, encouraging dietary diversity, new foraging strategies, and possibly even shifts in behavior that laid the groundwork for later human evolution. Living in Africa at the same time as Lucy's species was another early hominin called Kenyanthropus Platyops, who inhabited modern-day Kenya. This relatively flat-faced, small-brained bipedal species may actually represent a closer branch to modern humans on the evolutionary tree than Lucy's lineage. Before its discovery in 1999, the earliest known fossils with a flat face, a key shift in skull structure associated with modern humans, dated back only about 2 million years. Kenyanthropus, however, pushes that trait much further back in time. Its flat face could suggest a more direct connection to our lineage, possibly representing an early step toward the modern human skull. On the other hand, it's also possible that Kenyanthropus was an evolutionary dead end, and that flatter faces evolved independently in multiple lineages. Others in the scientific community believe more fossil evidence is needed before we can place Kenyanthropus within our family tree, as the only known skull from the species was found badly distorted. These scientists believe Kenyanthropus Platyops is no more than a variant of Australopithecus Afarensis. In terms of brain size, Kenyanthropus was similar to Australopithecus Afarensis, with an estimated cranial capacity of around 430 cubic centimeters. They had one of the longest known early hominin clavises, measuring 1.3 inches. Their nasal opening was narrower than that of Australopithecus, and their cheekbones were tall and sharply angled. Overall, the face resembles Homo rudolfensis, a species that came over 1 million years after Kenyanthropus. Due to the limited skeletal remains, its overall body size is unclear, though it was likely comparable in stature to other Australopithecines. This species lived in an environment that was a mixture of grassland and wooded habitat, comparable to the similarly dated sites of Laetoli, Tanzania, and Hadar, Ethiopia, where Australopithecus afarensis was found. While Kenyanthropus lived at the same time as Afarensis, Kenyanthropus's molars were smaller, indicating that the two species may have had different diets and therefore likely did not compete for the same types of food, though they were both probably largely plant eaters. Since our closest living relatives, chimpanzees, as well as other apes and monkeys, have been observed making and using simple tools, it is likely that all hominins made use of tools to some extent. But what truly sets us apart from the rest of the apes isn't just using tools, it's modifying them, especially crafting and shaping stone tools. So that raises the question, did any of these early hominins actually create and use stone tools? So far, no tools have been directly associated with the very early hominins, like Sahelanthropus Tchadensis, Orrorin Tugenensis, or Ardipithecus. Aside from the direct lack of stone tool evidence in the archaeological record alongside these species, a big clue comes from the limited number of hand bones found with these species. What we can gather from this is that while their hands show some features that might have allowed them to handle simple objects, they likely lacked the specialized anatomy needed for the precise and forceful grips required to make stone tools. Instead, their hand structure appears to have been better adapted for climbing and arboreal activity than for tool production. However, the Australopithecus species had hands that were well suited for the controlled manipulation of objects, and they probably did use tools. The oldest known stone tools are around 3.3 million years old and were unearthed in Kenya. These Lomequian tools, named after the site they were discovered on, were made from volcanic rock and crafted into flakes, which are a fragment of stone removed from a larger stone by striking it with another hard object or applying pressure. Cores were also found, which are the central piece of stone from which flakes are removed. It's essentially the leftover rock after flakes that provide valuable information about how humans made and used stone tools, and potential anvils, which are heavy, flat stones or rock fragments used as a stable surface to support other stones during the process of shaping these stone tools. Although Australopithecus afarensis is known from Kenya around this time, the most likely candidate for the toolmaker is Kenyanthropus Platyops, as specimens of this hominin have been found close to where the tools were excavated. Still, don't count Australopithecus afarensis out. There's compelling evidence they may have been among the earliest toolmakers. A small number of animal bones found at Dakika in Ethiopia have been reported as showing cut marks made by stone tools. They have been dated to about 3.4 million years ago, and the team involved attribute the butchery to Australopithecus afarensis, as this is the only species known to live in the area at this time. However, the conclusions are contentious. If they withstand scrutiny, this would be the earliest evidence of meat-eating behavior by a hominin. The first species of Australopithecus to be strongly associated with stone tool use is Australopithecus Garhi, who lived around 2.5 million years ago. Fossils of this species were discovered at the Bori site in the Middle Awash region of Ethiopia. Archaeological evidence from the site includes antelope bones that show clear-cut marks and signs of having been broken open, presumably by stone tools, likely to access meat and marrow. This suggests that Australopithecus Garhi may have been among the first hominins to transition to regular stone toolmaking and the consumption of animal flesh, marking a significant step toward behaviors later seen in early Homo species. This species is not well documented. It is defined on the basis of one fossil cranium and 4 other skull fragments, although a partial skeleton found nearby from about the same layer is usually included as part of the Australopithecus Garhi sample. Judging by this limited sample, it was similar in appearance to Australopithecus afarensis, but probably slightly larger in body shape and size and had more advanced features in the teeth. However, the premolars and the molars are larger than other Australopithecine and more similar to Paranthropus, a species of robust Australopithecines that are characterized by heavily built skulls capable of producing high stresses and bite forces. Garhi's brain size was about 450 cubic centimeters, similar to other Australopithecines. The associated fragmentary skeleton indicates a longer femur compared to other Australopithecus specimens. This suggests a change toward longer strides during bipedal walking. However, long, powerful arms were maintained. A changing climate had thinned the forests that once dominated this region, and savanna grasslands were becoming widespread. It most likely ate plant material, but if the antelope bones found at the site were indeed butchered by this species, then they must have included significant amounts of meat and marrow in their diet. Some scientists claim that the large molar teeth show that Australopithecus Garhi is related to the Paranthropus side of the branch. However, the combination of features of the face and brain case are unlike Paranthropus. The scientists who originally reported the finds think that Garhi may represent an ancestor of the genus Homo. The latest Australopithecus to live was Australopithecus Sediba, who lived around 1.9 million years ago in South Africa. The fossil skeletons of Sediba from Malapa Cave are so complete that scientists can see what entire skeletons looked like near the time when Homo evolved. Details of the teeth, the length of the arms and legs, and the narrow upper chest resemble earlier Australopithecus, while other tooth traits and the broad lower chest resemble humans. These links indicate that Sediba may reveal information about the origins and ancestor of the genus Homo. Functional changes in the pelvis of Sediba point to the evolution of upright walking seen in Homo, while other parts of the skeleton retain features found in other Australopithecines. Measurements of the strength of the upper arm bone and thigh bone show that Sediba had a more human-like pattern of locomotion than a fossil attributed to Homo habilis, a later hominin classified as human. These features suggest that Sediba walked upright on a regular basis, and that changes in the pelvis occurred before other changes in the body that are found in later specimens of Homo. For example, many paleoanthropologists maintain that the evolution of the pelvis in hominins was driven in part by the increase in the size of the head, and the pelvis needed to accommodate the birth of larger-brained offspring. But the reconstruction and analysis of 2 Sediba specimens' pelvises revealed that they had already developed certain modern features before brain size began to increase, and evolved primarily due to the demands of bipedal locomotion. The Australopithecus Sediba skull has several derived features, such as relatively small premolars and molars and facial features that are more similar to those in Homo. However, despite these changes in the pelvis and skull, other parts of Australopithecus Sediba's skeleton shows a body similar to that of other Australopithecines, with longer upper limbs and a small cranial capacity. The most remarkable thing about Australopithecus Sediba is that they are believed to have walked with a fully extended leg and a unique form of bipedalism involving hyperpronation. This means that when stepping, the foot would strike the ground on the outer edge, causing the foot to roll inward, with weight shifting to the inside of the foot before pushing off. While this gait was upright and bipedal, it differed from modern human walking, which suggests upright walking evolved on more than one path during human evolution. The combination of primitive and derived traits in Australopithecus Sediba shows part of the transition from a form adapted to partial arboreality to one primarily adapted to bipedal walking. Although some differences occurred between the Australopithecus species, overall Australopithecines were small, stocky hominins with a mix of ape-like and human-like traits. They had wide, shallow pelvises adapted for walking upright, though their rib cage and shoulders retained climbing features. Their brain size ranged from 400 to 550 cubic centimeters, closer to chimpanzees than modern humans. They had pronounced brow ridges, sloping foreheads, and projecting faces. Their teeth were intermediate between apes and humans, with smaller canines, thicker enamel, and larger molars suited for tough plant-based diets. Long arms and curved fingers suggest some tree climbing, while their legs and arched feet supported bipedalism. They lived in a variety of environments, from woodlands and riverine forests to open grasslands, though rarely in fully open savannas. Fossil sites like Kanapoi and Alia Bay in Kenya show they thrived in changing mosaic habitats. Their diet was primarily plant-based, including fruits, seeds, roots, and insects, with occasional scavenging of meat from predator kills. The genus Homo likely emerged from within the Australopithecines around 2 million years ago, but which species gave rise to humans remains debated. Fossils like Australopithecus Sediba show transitional traits, more human-like hands and pelvis, suggesting a possible link. In summary, Australopithecines were key players in human evolution. Their upright walking, dietary adaptability, and early human-like features paved the way for the rise of Homo and eventually modern humans. From Sahelanthropus Tchadensis and Orrorin Tugenensis to Ardipithecus and the Australopithecines, these early human ancestors lived across more than 5 million years, from around 7 million to under 2 million years ago. Slowly, through countless generations and gradual evolutionary change, they paved the way for the emergence of Homo. None of them knew the role they were playing, each individual, each family, simply trying to survive day to day, most likely without speech. And yet, their struggles and adaptations led to the humans who followed. Homo habilis, with their larger brains, dexterous hands, and long-term use of the Oldowan tool industry, to the pioneering Homo erectus, who harnessed fire, crafted Acheulean hand axes, and became the first humans to spread beyond Africa into Asia and Europe. Then came the more familiar faces in our evolutionary story: Neanderthals, Denisovans, and of course, Homo sapiens, who would go on to build complex civilizations and develop advanced fields like mathematics, astronomy, and physics. Another important field, paleoanthropology, has allowed us to uncover the lives of these fascinating ancient hominins, and, in doing so, better understand the deep origins of our own species. All of us, every human who ever lived, can trace our roots back to those ape-like ancestors who slowly left the trees and embraced life on the ground, setting the stage for the rise of humanity.